Hello. My name is Jonathan, and I'm from Israel. And I'm one of the members of the management committee, together with you took this time of me. Together with Dr. Stanley Freeman from the Agricultural Research Organization, that he will arrive this afternoon. So Israel is the Holy Land, and I mean Holy Land because it's land full of holes, and we put a lot of holes in order to monitor roots under various conditions, salinity, water stress, or excess of water, CO2, enrichment, temperature, and other things, wastewater irrigation. So we study roots in different various aspects, and using mini rhizotron and other facilities. We developed a new mini rhizotron, which is an automated one that go up and down automatically, and we can take pictures as much as you want. And this makes that also, we've developed new software in order to analyze the roots, convolutional neural network. So actually it automatically analyze the pictures and give us all kinds of information. We also work on monitoring roots under drought stress and use fluorescence proteins expressed under stress condition in order to see when the plants get into stress, in order to see how the plant behaves under different stresses. And the idea here is that the fluorescence proteins activate only under stress conditions. The group of Dr. Stanley Freeman is dealing with endophytes for increasing cucurbit plant performance by conferring abiotic stress tolerance. Drought and salinity are primary constraints to melon production in the Mediterranean region. And the fact that we cannot use methyl bromide anymore as a for fumigation make a lot of problems in all kinds of pathogens. And a system approach for improving melons crop production under stress-related growing conditions has been established by using novel endophytes isolated from wild cucurbits growing in similar conditions as in the Rift Valley in the south part of Israel, which is a very dry and hot area, about 35-40 degrees in the summertime. And the colonized endotype cultured melon plants are afforded tolerance to droughts and salinity conditions. So for summary, endophytes from wild cucurbits were isolated and colonized in cultured melon. Melons plants colonized with endophytes were protected against drought and high salinity conditions. In salinity and drought conditions, in field trials, 2 endophytes increased yield compared to untreated control, and experiments will continue to assess field performance of endophyte colonized melon in plants. Thank you.
Next presentation is for Montenegro.
Okay. Hello, my name is Jelena Lazarević. I am presenting Montenegro, and I'm working at University of Montenegro at Biotechnical Faculty. University of Montenegro celebrating during these days 50 years of from the time when it was established. But Biotechnical Faculty has a longer traditions, and before it was a research institute. Generally, I can say that in Montenegro we don't have really a research group who is working with beneficial microorganisms in agriculture. So I'm working in the field of forestry and representing Center for Forestry, and have a good collaboration with the colleagues in the Center of Soil and Melioration who are doing different type of research in soils. Regarding our institution, biotechnical faculty have I can say more or less typical organization. We are organized in 10 different research centers, but now I am presenting, let's say, activities in those 2 centers, which are more or less connected with our topics here. And here I can say what was research on the field of forestry and microorganisms. We were doing some research about mycorrhizal fungi, cultivation of ectomycorrhizal fungi, and inoculation of conifer seedlings. Those work was done in close collaboration with people, research group from University of Belgrade, Faculty of Forestry, because I get my education and postgraduate education at that faculty. After that, I switched to the study of fungal communities of root tips. We established good collaboration with the research group in Sweden in Department of Forest Pathology and Field Mycology, and we were focused on the study of mycorrhiza on high altitude pines. At the beginning, we were work on just on mycorrhiza on root tips, but later we were use metabar coding approach in study of fungal communities and in roots and soils. And what I would like to also say nowadays we started recently one project which is financed by our national funds, where we will going to research fungal biodiversity, but also we would like to promote the concept of soil health, and to work much more about microorganisms in soils, in agricultural soils, and to try to let's say raise public awareness about this matter. And so that's why we have those drawings here. Idea is that we will strongly try to support this approach. Thanks.
Next presentation is for North Macedonia.
Hello, everyone. My name is Natalia Tanasova-Panchevska, and I am from North Macedonia, MC from North Macedonia. Our Macedonian Root Benefit Network consists from 3 research groups. One is in Skopje, one in Prilep, and one is located in Štip. The first one is a research group from Department of Microbiology and Microbial Biotechnology from Faculty of Natural Sciences and Mathematics from Skopje. Here, I am a member of this group with 2 other participants, and we work on biocontrol of plant pathogens, antibiosis, soil microbiology, microbial ecology, and development of biofungicide and biosurfactant producing microorganisms, mostly on bacillus species, but also other soil microorganisms. Second research group is from PRILAB, from Scientific Tobacco Institute. My colleague is Biljana Jordanovska-Šiškovska, other MC from North Macedonia, and she is here also. They work on soil nutrients and pollutants and soil amendments, and mostly on Nicotiana tabacum, on tobacco. And the third group is from Faculty of Agriculture, University Goce Delchev from Štip, and colleague Daniela Todevska is she is working on horticultural production in greenhouse, seedling production, and also the impact of different soil microorganisms on this plant. So, thank you.
Next presentation is for Poland.
Hello, my name is Joanna Banasiak, and I have pleasure of presenting the Polish Root Benefit Network that currently comprises 12 research groups from various cities, and due to time limitation, the presentation will be structured around selected topic related to our action. So the first one that is close to my expertise is the role of active transport system in Legume Rhizobium symbiosis and arbuscular mycorrhiza. And this research is conducted by two groups from Institute of Bioorganic Chemistry, Polish Academy of Sciences, Poznań, and Poznań University of Life Sciences. The group led by Michał Jasiński, my boss, is focused on ABC transporters and their role from Medicago truncatula, and their role in symbiotic interactions by translocating plant hormones, particularly cytokinins, but also signaling molecules and secondary metabolites translocation. Karolina Jarzyniak recently initiated studies on plant membrane transporters from another model legume plant, Lotus japonicus, and the involvement on plant membrane transporters belonging to multi-drug and toxic extrusion proteins and their role in symbiotic interactions under drought stress. And these studies are related to working group 2 and are conducted in vitro, climate chamber, and glasshouse scale using molecular biology approaches, phenotyping, of course, transport experiments in different heterology system, HPLC-MS, and confocal microscopy. This next topic, well recognized in Poland, concerns plant microorganism interaction under metal toxicity. And here Piotr Spondek groups from Małopolska Center of Biotechnology at the Jagiellonian University specializes in researching microorganism dependent adaptation of plants to living in metal-contaminated conditions. Katarzyna Turner groups, also from Jagiellonian University, is focused on plant symbiotic association with microbes, and both mycorrhizal and endophytic fungi, as well as rhizospheric and endophytic bacteria in extreme habitats, including toxic metal-polluted environments. She's also interested in microbial-assisted phytoremediation. And this topic is also part of the Marzena Sujkowska-Rybkowska work at Warsaw University of Life Sciences. She focuses on legumes. And here again, molecular biology, chromatography, and microscopy is used, and notably core facilities of structural biology, both crystallography and cryo-EM is applied and available in Małopolska Center of Biotechnology. Another topic widely explored by Polish research groups is microbiome and the utilization of microorganisms in crops. And here a lot of groups are interested of this topic from various cities: Toruń, Lublin, Łódź, Skierniewice, also Poznań and Krakow. And these groups often closely collaborate to investigate beneficial microorganism diversity, dynamics of plant microbiota in response to different environmental stresses, and remodeling a plant microbiome. At least part of them is also interested in selecting of endogenous microorganisms for use as a plant growth biostimulants. And the research presented in this slide addressed the interest of all working groups, and in addition to molecular biology, in addition to in vitro climate chamber and glass house, they use also field trials, are conducted in a field tray scales. From my side, so thank you.
Next one is for Portugal.
Good morning. My name is Isabel Brito. I'm coming from Portugal, and I will present the main laboratories or institutes that are working with soil microbes in general. They are split all over the country, mainly in the littoral parts, and of course more in Lisbon, but also in Porto. In Coimbra, it's a group that is not very active right now, but it has been some years ago, so they still have people working on the subject, but not very intensively. Aveiro, and Évora, where I come from, and Elvas. So what I did was a large survey and pointing the groups that are working with these microbes, our cost working groups where they develop the work they develop fit in, and the person responsible for those groups. So the Faculty of Sciences, and another detail, research institutes are very connected with the faculties or the universities, so when we have both in here, it's because this is Faculty of Sciences and this is the research institute that goes together. In here we have the group of Cristina Cruz that deals with plant soil, is the plant soil ecology group that works mainly with rhizobia and arbuscular mycorrhiza, but also other bacteria, and is one of the oldest groups working with this with soil microbes, and addresses their work fits in or addresses the objectives in all of our 4 working groups. Also in the Faculty of Lisbon, the Agronomy Institute has a plant and the group of plant science and crop production that Luís Rolão is in charge, and they developed work mainly on what is related to our working groups 3 and 4. Also in Lisbon, Greenit, it's an institute that puts together a lot of researchers from top institutes from Lisbon. So in fact, people from ITQB, Gulbenkian Foundation, IBET, INIAF, so it puts together a lot of researchers, and they have great expertise in studying of microbes in general, so, and they also address soil microbes, and particularly in the group of healthier plant nutrition for food security and quality, which Margarida Oliveira is in charge. And so in here I included for the 4 working groups, because in fact they have a very strong expertise in the different aspects of microbial interactions and so. Also in Lisbon, INIAV, one of the oldest institutes working with soil microbes, particularly on Rhizobia. They have one of the greatest collection of Rhizobia strains in our country, so this is an important thing when we are working with this bacteria. Sometimes we want other strains and we want to compare and know about where are the collections. And in fact, they are connected with international entities also that harbor great collections. In here, Isabel Videira Castro is the person in charge, and so a group mainly addressing Rhizobia issues. Then in the northern region, we have, I lose the mouse here, Catholic University that is connected with the Biotechnology Science School. Particularly the group is Centro de Biotecnologia e Química FINA, Biotechnology Center and Chemistry. The person in charge is Sofia Ferreira, which is going to have a presentation this afternoon, and the aspects they deal are mainly connected to working group 3 and 4. Also in Porto, there's another group dealing with benthic ecology and environmental solutions. That doesn't seem to have a big relation with our organisms, but in fact they have a group that work with some bacteria and inoculum production or coming from algae sometimes. So connected with working group 4. Okay. In the University of Aveiro, again, mainly focusing on Rhizobium, the biology department led by Telvina Figueira, and our group in the University of Évora in the south of Portugal. We are connected to MED, it's our research institute. We deal mainly with arbuscular mycorrhiza and also other bacteria, including Rhizobia, and we can include our work in all the working groups that fit in this cost action. Regarding arbuscular mycorrhiza, we work mainly with native inoculum, and the approach is how can we adjust cropping systems in terms of the cropping practices to make the best use possible of the native populations, and also the interactions between arbuscular mycorrhizal fungi and other bacteria, not only Rhizobia, but also other soil bacteria. Our model plant for Rhizobia is chickpea, which is not very common in more northern European countries, so we do have a very big collection of chickpea Rhizobia. And also in the south region of Portugal, the Innov Plant Protect Collaborative Laboratory, which is a collab that gathers a lot of other laboratories and also has some work on these aspects, mainly relating to working group 1, 3, and 4. Thank you.
Next presentation is for Serbia.
Greetings to everyone. My name is Jelena Jovičić-Petrović, and I will present Serbian Root Benefit Network. 4 institutions replied to our survey. 3 of them are located in Belgrade, and 1 is in northern Serbia in Sremska Kamenica. The first institution is Faculty of Agriculture. I'm coming from Faculty of Agriculture as well as our co-leader of 4th working group, Vera Kalajic. We work on plant growth promoting bacteria and biocontrol agents from different genera, mainly Bacillus, Azotobacter, Pseudomonas, Halomonas, and Trichoderma, and with different plants of interest depending on the current project, but mainly cereals. Our research includes steps from isolation, characterization of mechanisms toward application, because we collaborate with industry and We work on consortia design, and in recent years we are applying encapsulation techniques in order to provide better shelf life and better survival of microbes. We have conducted multiple projects related to use of plant growth promoting microorganisms in different areas such as agriculture, horticulture and forestry, and bioremediation, all aimed to improve plant response to abiotic and biotic stress. And here you can see some current research topics that include halotolerant plant growth promoting bacteria, isolated from rhizosphere plants growing in naturally saline soil in order to be used in agriculture, then seed biopriming, the effect of beneficial bacteria on germination and early growth stages, biological control, development of biofungicides and bioinsecticides. And the last topic we started last year is biofortification, trying to get multifunctional use of trichoderma, for example, to enrich crops with selenium-enriched plant growth promoting trichoderma. Next institution is the Biofor System Company. Their representative, Mr. Bogdan Žikić, is here today as another MC member. It is a Serbian company producing microbiological fertilizers, soil conditioners, and biostimulants, mainly based on nitrogen-fixing bacteria, and representatives of Bacillus, Pseudomonas, and Trichoderma. And the preparations are produced and packaged in the company's facility and intended for use in conventional and organic agriculture. They provide significant support for their end users, for example, organizing different educational events or annual field trials. And besides that annual field trials, they are developing new strains, expanding their collection, currently working on biological control, all that thanks to the multidisciplinary team and collaboration with Faculty of Agriculture. The next institution is Faculty of Biology. They work on biological control and plant microbe interaction. They are focused on Bacillus and Pseudomonas. And they work with Bacillus and Pseudomonas strains as biocontrol agents, antagonists of plant pathogens, and nematocidal effects. For example, nematocidal hydrogen cyanide-producing Pseudomonas strains. They study phyllosphere and rhizosphere of plants and searching for beneficial plant growth promoting strains that can be used to improve resistance to drought, and they started new topic on type 3 secretion system in plant growth promoting bacteria and its role in plant microbe interaction. And the last institution is Educons University, located in Sremska Kamenica. They are focused on biological control, mainly trichoderma isolates, and plant microbe interactions. Plant of interest is tomato. So they are mainly working on plant expression in plants. Thank you.
Next presentation is for Slovenia.
Hello everyone, my name is Irina Maček, I come from Slovenia. So here at this presentation I'm presenting University of Ljubljana. To the survey we only got these responses from 2 groups, but now similar to other countries I think I should be more intense in contacting specific persons who I think will reply if we do it directly. So there are 3 universities in Slovenia basically. It's a small country And I think at the Coastal University also, they are working with Olea, oil trees, so, and the University of Maribor, which is more to the northern eastern part. There is also agricultural group, so I think this would be more like not directly, but indirectly they would be interested in these soil beneficial organisms. For Ljubljana, there are two departments where there is quite a lot of focus on plant-soil interactions and also beneficial organisms. And we are also collaborating together. So from Department of Biology, it's my group. We are ecology group. And from Department of Agronomy, there's a chair of soil science and environment, and there is Professor Marietka Suhadolc, who this time could not attend. And they are also working on soil beneficial organisms. And like I said, we are collaborating together. I have just recently moved to biology department 2 years ago. Before that, I was also working at the agronomy department. The topics which we are interested, so in my group, there has been quite, for a long time, focus on arbuscular and mycorrhiza in different ecosystems. So I cannot really specifically say it's one plant or one that I would be more interested in, but it's in general crop species or horticultural species in different management systems and also in indigenous and invasive species. But the focus is on arbuscular mycorrhiza. And that is from the point of different abiotic stressors which affect the community and mainly the community ecology, but also other interactions, and together with the plant part, plant ecophysiological responses. And the systems go from in vitro cultures from lab techniques to glass houses, and we have, together with the group of agronomy, quite a lot of field trials, quite good capacities also at the, around the university and in different other areas on field experiments. Some of them are also long-term. And one also thing I did quite a lot of research on is on extreme environments that provide a nice system. So these are natural CO2 springs, which are also located in Slovenia, which provide a nice system to do research on ecology of different organisms. And then the second group from Marietka Suhadolc, they have quite extensive knowledge and equipment on soil, all aspects of soil science, and they manage several long-term experiments also with minimum tillage or different practices, different fertilization, and with different species. Metka specializes quite a lot on nitrogen cycling and different aspects of that, on functional genes and different, with the different crop systems, as I said. And that would be all. Thank you.
Sorry for taking the space of Spain here. So my 12 points go to, no, sorry, it's another contest. Good morning, everyone. I'm happy to represent Switzerland. Sadly, I'm the only representative for Switzerland at the moment, so I hope this will change for the next meeting. I'm also involving people in here, so it will be hopefully filled by the next meeting. I'm actually from the Zurich University of Applied Science, and I mainly had 3 answers to the survey to present you today, but I bring also here, it's a part of the bottom of my slide, some possibilities of other partners to join the cost action. In the figure, you have also the Zurich Basel Plant Institute, Plant Science Center, sorry, which is very important, and that's mainly the 3 partners that I'm going to present today here. We have in Switzerland, like other partners, universities of applied science, like for example in my case, and also ETH and EPFL institutions. Also, we have like equivalent from INRAE in France, Agroscope, FIBOL, and other competences center in Switzerland. So concerning myself, I'm from the Zurich University of Applied Sciences. I'm actually not in Zurich, but close to Zurich, and I'm in the group from Environmental Genomics and Systems Biology. I'm interested in dysbiosis, symbiotic interactions, and mainly omic technologies. Concerning the plants of interest, we mainly work with plants that we can handle easily in the lab, because we don't have really a big facility, and we work very often with biosafety level 2 organisms, so we work with lettuce and strawberries, for example. But we are also well known in our group from Malus and Rubus species, and we also, or at least personally, have a personal interest with leguminous crops, and I submitted a project to fit in this cost action. That's a specific call from our national funding agency to fit in within cost actions regarding leguminous crops and soybeans. So it was last Friday, fingers crossed that it will be accepted. Concerning the other person that answered the survey, I contacted Marcel van der Eyden, and he's also willing to act as an MC member. Let's see if he applies. But he's more focusing on AMF with crops, vegetables, and herbs. He also mentioned he has interest for bacteria and fungi, but as far as I know, he's mainly into AMF, and most of you probably know him already. Another person that answered the survey is actually Pascale Fleury from the University of Basel. She works more on plant microbe interaction, especially with Pseudomonas protegens, so bacteria that are benefic for the roots. She works also with Phengomonas, Pythium, Rhizoctonia solani, and others. And she's working on crop plants and also on Arabidopsis as a model. Finally, as I mentioned in my first slide, I will also present you possibilities to extend the network. So in the University of Zurich, I'm in close contact with Cyril Zipfel, who would be interested to join, but he doesn't have project in this direction. I also mentioned Beat Keller is going to retire pretty soon, but is actually going to be replaced by Cyril, so actually there will be here connection regarding the plant aspects. At the ETH of ETH Zurich, I identified Monica Marofor, Bruce McDonald, and also Bruno Studer. Bruno Studer is more onto the plant side, whereas Monica and Bruce are more on the bacteria and fungi, respectively. The University of Basel, I will also contact Klaus Schleppi in order to join us. He's active in plant microbiome in general, so this would be a great addition. We have the University of Fribourg, where Laurent Falquet and Laure Weisskopf, where they could be also involved. The University of Neuchâtel, we have Daniel Kroll, who mentioned already his interest for another cost action. And the PFL Lausanne, Christophe Kiel is going to retire soon. Maybe there's somebody taking his position afterwards. And the University of Geneva, I think there is a little bit less, it's more to the plant side, but at least I've identified Roman Hull and Marie Barberon, who could be actually interested to join. That's it from my side. Thank you very much.
Next one is Spain.
Hello everyone, my name is Estíbaliz Larrainzar and I am representing the research related to this action in Spain. As you can see, also here from the audience, we have quite some, this topic is quite studied in Spain, and we have institutions all over the country. This big square is Granada, which is one of the highest, highest number of research groups that are fitting in this action, followed by institutions in Castilla y León, Madrid, and then Navarra, where I come from. Then we have some other regions in Valencia, Aragon, Catalonia, and Galicia. So, as you can see, a bit over the place. In my presentation, since the number of groups is so large, I am not going to be able to focus on the details of each group, but as you know, the Excel file of the survey is going to be distributed, so you will have the details there. So what I did is I just grouped it in the 4 main topics that are covered in these research groups. The largest by far is the one addressing beneficial plant microorganism interactions, and you can see there are institutes all over the place, and they address a very large also variety of crops, ranging from model plants like Arabidopsis to other horticultural species, and also trees, so woody species and aromatic plants. And what they do analyze the different plant growth promoting bacteria. They also try to understand agricultural management practices, the forest microbiome, stress tolerance, and also some fungal microbiome of plants and root fungus interactions. So those are the main topics. And the range of techniques is also, it varies from omic approaches to more bioinformatics, molecular biology, and again, from field experiments to more in-plant studies in the lab. The second largest topic will be the one addressing legume rhizobium symbiosis. This is where I fit. So we have here representatives of the Estación Experimental de Auladell in Aragon. Manolo Becana is here. And I am coming from the Public University of Navarra. And this institute, it was not in the survey, but I would like to mention that it's a public institute that carries out field experiments, and they have direct contact with agricultural end users, final end users. So it's really an interesting, let's say, incorporation in the action. Actually, they were supporting the action from the beginning when we submitted. And again, in Granada, we have representatives from the university and the Estación Experimental del Zaidín, which is part of the National Research Council in Spain. So this is where the largest group of researchers is. And again, here the number of techniques and topics varies from the molecular details of the symbiotic nitrogen fixation, some work on the nitrification and nitric oxide emission, and then more developmental and stress responses in nodulated legumes. And then again, of course, this covers a range of legume species as well. Then the third topic will be the arbuscular mycorrhiza symbiosis, and here we have representatives from Catalonia, Valencia, and again in Granada. And the topics are mostly about sustainable plant protection, plant immunity and biochemistry, and also biofertilization and bioremediation using these fungi. And this again covers a range of species from woody crops to more horticultural crops. And they do a number of tools. They approach using a number of approaches: metabarcoding, microbial isolation, in vitro and in vivo cultures, and more classic molecular techniques. And finally, the let's say representative topic will be the plant microbe pathogenicity. Sorry. So more about pathogens, right? So not so positive, let's say, interactions. But they are also working on in different regions. Here, the most representative is the one in Madrid. And they work on, again, also agricultural management and more forest pathology and endophyte interactions. So I am running out of time, so that's it. Thank you very much.
Next will be Turkey.
Hello everyone, I am Ersun Atay from Turkey. We have shared our surveys with the colleagues from universities and institutions in Turkey. So far, we have received some answers from research groups in 17 different cities in Turkey. On the screen, you see the names and locations of these cities. I'm sure the more the action gets known and the more stakeholder will be represented here from Turkey. So far, Izmir has been in the foreground. 3 different institutions and 3 different research groups at 3 different institutions in Izmir are in contact with us. I am one of the 2 members of Turkish Management Committee in this action. I have been working at Burdur Mehmet Akif Ersoy University in the Mediterranean region of Turkey. We are working more on the effects of plant growth bacteria on plant nutrition. We work on fruit species, especially apples and almonds. And we do our research in the field also at the molecular level. The other member of our management committee, Onur, and is at Koç University in Istanbul. They are working similar topics with Stuart, and they use wheat, potato, and some fruit species like hazelnut in their research. The Middle East Technical University has a team specializing in molecular biology, and they use field crops mainly. And also we have another team in one city of Turkey, and they are studying on phenotyping and molecular biology. They are investigating some interaction between plants and root-associated bacteria in their labs. Also, there are some teams in some different universities in Turkey, and they are in contact with us, and they use different types of plants in their studies. Also, they are doing research and how bacteria affect the plant growth in different parts of Turkey. In Turkey, we have a large organization and focused on agricultural research. And this organization called TAGEM houses more than 50 research institutions in Turkey, and they work on all plant species grown in Turkey. And some of them are in contact with us, but some of them did not reply our surveys. And they do some research, especially some groups working on horticultural crops and field crops are doing a lot of experimental research in the field, also in the laboratory, and some of them are isolating some certain bacteria. And so far, nearly 20% of the members of this action are from Turkey. And this is high rate because our organization called TÜBİTAK did good advertising to do this. And also, as I said before, TAGEM is running a lot of research in agricultural area, and some of them did not answer our surveys. And 3 universities, and Yeditepe in Istanbul, and Atatürk in Erzurum, and Selçuk is in Konya, are doing good research using bacteria, but so far they didn't answer our surveys. And big 3 companies called Integro, Gübretaş, and Toros Tarım. These companies now have initiated some investment to produce some biofertilizer, and they are pursuing good research to achieve this aim. Thanks.
So it's UK.
Okay, so hi everyone. It's indeed a pleasure being here and see so many different options for collaboration. Let me start with two disclaimers. So the first one is that similar to what also other colleagues have mentioned, so consider this the very first iteration of the survey. Some of the people I contacted did not reply, and I chased up with them, while maybe other groups that are working with plant microbe interaction and probably with root beneficial microbes were not included, and we will correct for that as well. And the second disclaimer is that I'm the only MC representative for the UK, so we are looking for another one joining us. Now, so the four groups that responded to the survey are myself and 3 colleagues in England, so I decided I'm going to go a bit into the details of what they are doing. And I structured this presentation with the target organisms, so either the plant or the microbes they are working with, the scale of the experiments, and basically what they are doing. In my group, we are interested mostly in bacteria, thriving at the root soil interface. Our favorite model, plant model, is barley, which is a crop. But at the same time as this, we consider a very interesting feature that wild ancestors are accessible for experimentation. So this allows you to investigate plant microbe interaction within the framework of domestication, so something that you can't do with model plants like Arabidopsis thaliana. We go all the way from field trials, actually real farms, to more reductionistic approaches in axenic environment, although most of our work is glasshouse based. Our interest is in how plants and plants genetics shape those communities, so we are very active in cultivation independent survey of microbes and the genetics of the plant. In Cambridge, Jiong Ming group is primarily interested in arbuscular mycorrhizal fungi. They work with cereal as well, mostly wheat and rice. Their type of work is more on the basic side of the interaction, so therefore is climatic chamber and glass houses. And they are interested in the genetics, in particular the genetics of the host, so how the plant regulate the symbiosis. So at University of Warwick in Coventry, the group of Miriam Gifford is interested in legumes and mostly the model Medicago truncatula and rhizobia, as well as other bacteria that collaborate or interfere with rhizobia in establishing nodulation. They have also a range of scale that goes from field trials to more reductionistic approaches. So I basically copy-paste what are our work. So imagine this is a legume, not a cereal, but that's the same scale. And similar to what we are doing, they are interested on how plants shape those communities and what kind of impact this may have for the plants, and the genetics of the host, so mutants in Medicago that may again contribute or interfere with these processes. Then the John Innes Center in Norwich is probably one of the, in terms of research institution, one of the more active institutions working with plant sciences in the UK, certainly the one that may profit for the better funding. Matt Hutchins' lab is working with bacteria and predominantly with Streptomyces. Their interest is again cereal species, so wheat and barley. The scale of their work is again climatic chamber, or how should I say, competition assay between members of the microbiota in vitro, so in play, microbe interaction, so more on the basic side of the work. Similar to other groups, there is an interest on how plants may modulate those microbial communities and then monitor their dynamic at the root soil interface and the genetics of the plants. So the John Innes Center has a very well-developed plant transformation facility. So if you are interested in, for instance, genome editing in crops like wheat and barley, I would strongly recommend to approach them. And they've also the opportunity to work with a stable isotope, so tracing who is producing and or eating what, and that's again could be an interesting avenue for to explore. As may have already seen from these slides, each one of them is willing to accept visiting students or junior scholar as a STSM. I know all of them and I can vouch for them, are very decent people, very smart scientists, so please interact with them. And that's all from my side.
So we will have the second part for Albania, which was not combined between the two NC members. Since we are on time, we can do it.
Hello to everyone. I'm Suela Spahiu. I'm from Albania. I represent my institution, that is the Center of Agriculture Technology Transfer. This is an institution that is opposed to the Ministry of Agriculture and Rural Development. The mission of the Agriculture Technology Transfer Center is a state institution for research, transfer of technology in field of agriculture and rural development. It serves the current and prospective development of the country agriculture and in accordance with and in implementation of the strategy and policies of this sector, which are the main objective of this transfer center. One of them is generating adaptation, testing, and transfer of agriculture technology in function of technology and technology progress, increase of efficiency and competitiveness of agriculture and livestock production, sustainable development and environmental protection. Second one, these centers provide expertise, training, and information and agriculture advisory and agribusiness structure. Third, providing expertise, training, and information for agriculture advisory and also, as I mentioned, agribusiness structure. Main object is land and water service. This center, we have the head of the Transfer Center of Agriculture, that is upon of land. We have the director of land and water service director, that has two sectors, the sector of the water and GIS sector. We have the directory of livestock technology, and that is which has two sectors, the sector of breeding technology and the Russian lab sector. We have another sector that is important, integrate farm management, and also we have the laboratory sector that has the laboratory of biologic materials, with the laboratory of soil and water, and the fertilizer product laboratory. We have also the experimental and manufacturing-based sector, which in 2024 testing 5 variety of barley. We have testing of 30 wheat variety. We have also testing of 5 alpha variety, which I'm responsible in this institution. Also, at the end, we have the finance sector of institution. To represent myself, I have a PhD from Agricultural University of Tirana in plant protection. Y has obtained several certificates from post-grade courses. I'm actively participating in workshops and conferences related to agriculture and environmental issues in Albania and abroad. I'm currently head of the seed sector in Agriculture Center of Technology Transfer in Tirana, which is located in Fuscru, Albania. I work previously in private sector in consulting company, and also I'm a professor in university. I represent Albania as a member of management committee in 3 action of coast, and as well as in the regional rural development as expert of climate change, also external expert of General Directory of Accreditation, and I'm act in the past as external expert of Quality Assurance Agency in higher education. Thank you very much, and I would like to thank you for the invitation.
Okay, so we have finished with the presentation by the MC member who are here today. There was a presentation sent by Ireland, but there is no MC member, and I don't feel enough confident to present their network. So anyway, I will distribute the presentation, and also the survey from Ireland will be in the final version distributed to participants. So thank you everyone to keep on time. We managed. It was a bit risky to have this presentation by all countries. What I noticed is there are many possibilities of creating consortia by complementary expertise. I think we are very strong in microbiology, inoculant production, and use in plant physiology and in agronomy. What I see, maybe it's a bit less strong in our networks, is genetic, except maybe in UK where it was stressed out. And I think that's a point we wanted to be included, to make co-selection of microorganisms and of crops. So it's important also we try to attract some geneticists. Probably also in soil science, there were some expertise which were presented, but we are a bit weak on this. So we should try also to attract some soil scientists. And also we, in the working group 3, we put an aspect on the economy of this beneficial plant microbe interaction. I don't think we have any economists here today. Some answered or applied to the working group, so they could participate, but I think it's a bit difficult to have them in each meeting. Probably we can have some specific workshop maybe on this topic and try to invite them for such topic. Okay, so I think now we can finish with this session, having lunch, and start again at 2 p.m. here. If there are some presentation, we still need to be uploaded for this afternoon presentation. Please, can you come now? Yes, and we will make a group picture, maybe here. No, so, yes, you are here. So can we make a picture of everyone here? So maybe we can go here, just in front before the lunch, in the hall.